All right, this is part two of the genetic testing topic. So the first we just talked about genetic testing and etc. However, now we're going to go into what I call the molecular biology of proteins, of the genes, and what do genes do? They are the code for making proteins. And how is that done? And that's what we're going to be talking about in this particular topic. So as you can imagine, this is part two, so this is going to be probably another lengthy video. So hopefully, as you did in the first one, I'm assuming taking them chunks. It's a lot. It can, you need lots of coffee for one to listen to my monotone voice. But there's a lot of good information here. I definitely encourage you looking over the videos because the videos do enlighten you a little bit over what you see and what I say. It's nice to have an animation. And I've got two videos right off the bat. One is dealing with the genetic engineering. It's just an animation about what if and what are the possibilities. Today, you might say that's kind of silly, but tomorrow it very well may be here. And then this, a lot of you may have heard this about CRISPR, which is at the bottom here. And that is an actual technology that is being used that is very, very powerful in solving and potentially solving a variety of diseases and such, and changing how we live and the longevity of how we live. So anyway, so this is a very good video, just kind of giving you an explanation of what CRISPR is, and also a lot of the background information that you will have already learned, and it will hopefully make sense to you. That's my hope is with this class, is for at least, not that you're going to be a medical doctor, but at least you have a reference of like, hey, I think I know what he's talking about, or she is saying. Okay. So anyway, something to check out for you. By no means is it for a test or anything like that, but I just like to put these as supplemental for those of you that are curious, that want to learn more about it. Anyway, all right, so let's go. What we've got here, I've just got a little notation of the structure of DNA, which we already know. We'll go into it a little bit more, how DNA is replicated. We're already familiar with the cell cycle and mitosis, and we know that the cell cycle in the S phase of interphase is replication. Well, how does that occur? So we're going to take a look at that. And then we've got transcription and translation, which involves a couple other players and organelles, hence is why I have this down. So you can start seeing that you don't have to necessarily memorize all of these organelles. We're just putting them into the biological processes of functionality. Okay, we already know the nucleus holds the DNA as an example. Vesicles are trash cans, when we learn with endo and exocytosis. The mitochondria is with cellular respiration we'll be getting into. Ribosomes is going to be talked about now. That's your contractor that assists in creating proteins, okay? And, about cytoplasm, plasma membrane when it comes to diffusion, letting molecules in and out. So you're going to know all of this stuff. That's a piece of cake. All right, so let's move on. Okay, so first of all, obviously we're talking about eukaryotic organisms. We're talking about DNA in a nucleus and the fact of replication. Then we are talking about eukaryotic organisms. Of course, we're talking about protists, plants, fungi, and animal cell. And then we're going to be talking about what is DNA and how is it packaged. Now, remember, and it gets very confusing, and don't feel like I can't believe I don't understand this. DNA has different names for its different looks or condensations. It kind of like, I think I used this example before, it's like a street that goes through Pensacola. Same street, it has 4 different names. I don't know, maybe is it Mobile Highway or Michigan Avenue? I forget. I don't know. But I know there's streets like that change names, and DNA is one of them. DNA is a 5-foot molecule. As it condenses and wraps around histones or proteins, that's what helps it do the coiling process. Okay, now you got a chromatin. All right, and then when it starts really condensing into a form of what we know as a chromosome. Still the same thing, it just has a different form. It's much more condensed. Okay. And then, of course, we're going to take a look more closely at our nitrogenous bases, which, of course, is the form of the genetic code. Okay, that's what forms our genes. That's code that within the genes forms our proteins. Okay, so let's take a look at it. All right, let's just compare and contrast our prokaryotes with eukaryotes in replication. The prokaryotes, circular DNA. Okay, what is that circular DNA? You should already know this. Those are your plasmids. You can see it's much smaller. Okay, and only has one origination point, meaning there's a beginning and an end. Okay, with eukaryotes we have free ends because it's linear, it's not circular. Then when it comes to the haploid cells, well, of course, haploid, you should know what the haploid is, half. So you're talking about your sex cells. Okay, it's about 1 meter long. Lagging strand is not completely replicated. You might wonder what is the lagging strand. Well, you won't know yet, but you soon will. And then small pieces of DNA are lost in every cell cycle. This is part of the reality of aging, if you will, as well. Okay, so think about this. Every time DNA replicates, we lose a little bit of integrity. Okay, and our DNA replicates depending on what body part, over and over, like our skin cells do. They have a high frequency of replication. An analogy would be you got a copy of something. You know, say you just created a nice piece of art, or you just typed up something, you printed it out. That's your master copy. Now you take that copy, put it on a copier, and you make a copy of that. Then you take that copy that you just made. That's now the new master, and you make a copy of that. Do that thousands of times. Eventually, that 10,000th copy is probably going to be faded. It will lose its crispness. Similar concept to DNA replication. And then we won't go into great detail because now we're talking more into biochemistry, but we have end caps or telomeres that help protect or retain genetic information. So even though our DNA ends become unraveled a little bit when it comes to replication, these telomeres are like masking tape that secures the end so it doesn't completely come unglued. But we still do lose a little bit. But again, that's biochemical stuff. That's way down the rabbit hole. We don't need to get too into detail on that. So just understand the difference between prokaryote and eukaryote. Prokaryotes are circular DNA. Only one origination point. Eukaryotes, which is us, we have free ends and more complex. That's about it. Okay, the anatomy of DNA and RNA. Okay, make sure you know what DNA is, deoxyribonucleic acid. And that is made up of 4 kinds of bases. Make sure we know what they are. DNA. Make sure you know cytosine, guanine, adenine, and thymine. Okay, those are your 4 nitrogenous bases. Okay, and then we've got RNA. Now notice RNA only has 1 backbone. Okay, and we'll talk about how that is important. And then we've got the same thing, cytosine, guanine, adenine, but notice that uracil does not, or exists in RNA, but does not exist in DNA. Uracil is a placeholder. It takes place of thymine. So that is the molecular difference between RNA and DNA. Okay, this is the actual record of, say, this whole section right here is a gene. This right here is created. This is the reading of the gene. Okay, as an example. And we'll get into that a little bit later on in more detail. But you could see with this backbone is the same as this backbone here. So these are identically the same. It's just that there's 2 of these backbones and only 1 here. Ribonucleic acid. RNA is nothing more than a messenger. It's a molecule that reads the DNA for whatever gene that needs to be read to create a protein, whatever the cell needs. And then this RNA then gets read or translated. Okay, so this code that got transcribed from the DNA, okay, which of course happens in the nucleus, then floats out of the nucleus into the cytoplasm of the cell, and that's where your ribosomes take hold, and then actually creates a new molecule, okay, and that ends up being a protein. That's the Cliff Notes version of it. Some would say that's the central dogma of biology right there. Okay, so let's make sure we understand Chargaff's rule. Super simple concept. The concept is that your molecules, adenine and thymine, always pair together. Okay, guanine and cytosine always pair together. Always, always, always. If for some reason adenine pairs with cytosine or thymine pairs with guanine, then there is an error. Okay, these two always match up. Don't worry about the purine and the pyrimidine. These are just the labels. Adenine and guanine is a purine. Thymine and cymine is a pyrimidine. But we're not going to worry about that, okay? But what Chargaff's rule means, what it is that if you think about it, you're looking at a gene, okay? And through experimentation and such, it is determined that you have got 20% of adenine. Okay, that molecule is comprised of 20% of this particular molecule. So if you know that adenine always pairs up with thymine, then it would make sense that 20% would be consumed of thymine. They should balance out. Okay, now let's go one step further. So that's 40% of the whole, because we're looking at the whole thing is 100%. So you've got 20%, that's 40, so that means that this is going to be 30% is going to be guanine and 30% is going to be cytosine, approximately, more or less. That is Chargaff's rule. The amount of adenine is equal to the amount of thymine, and amount of cytosine is going to equal the amount of guanine, always in equal proportions to make up the whole. Make sense? Okay. And notice that the adenine and guanine, look at these are larger molecules. Okay. And again, there's lots of illustrations. They're all over Google. You don't have to depend on this. You can Google it yourself for better reference, or just to figure out what's going on. But here's your backbone right here. That's your backbone of RNA or DNA. And then you've got a sugar right here. Okay. This is your sugar. This is your ribose, part of a macromolecule. And then your nucleotides, these are bonding to that. So here you've got, it could be, well, I guess, well, it says it, adenine. This is guanine, okay? Then you've got cytosine and thymine. And then on the other side, as we'll see when it's illustrated, I got other illustrations, you'll see that what binds to adenine, then thymine will be on the other side. Matter of fact, I think the next slide may have that. Okay, so let's go ahead and go to the next slide. Okay, so now we are looking at a couple of things here. We're looking at what DNA looks like at various levels of condensation, meaning condensing. And again, just another illustration showing the anatomy of the DNA. So this is something, there's some things here I want you to know. So here you've got DNA that is coiling, this is regular DNA, that is coiling around, they're like little golf balls. They're called histones. These are proteins, okay? This just helps with the condensation process. There's more to it than that, but that's all that we need to worry about. And then, of course, this is just showing a section of that coil. And then I'm going backwards, okay? And you can see we start off with the chromosome, so basically it's decoiling. But all these, this is all DNA in different shapes, but it just changes name. It doesn't reference it here, but the names change depending on what you're looking at and who's illustrating it. Okay, but histones, another name they'll call this whole feature is a nucleosome. Okay, I think I've got a slide for that. And then, of course, when it's fully condensed, that DNA is now called a chromosome. But just know that these little, also another name for them are bobbins, but definitely histones. These are proteins, just assist in that coiling process. And then here's just another illustration of your backbone of DNA. It's double helix. Here's your base pairs. Your lines are your hydrogen bonds. Okay. And again, these are just another illustration showing that, hand in glove. These are more geometric than curved. So obviously, if this tried to bond with this over here, it wouldn't work. This one does not have the same shape as this one. Okay. All right, just more illustration. I'm very repetitive in I get carried away in seeing all these illustrations. Let's see, is there anything fascinating here on this slide? Well, here's that bobbins. Wound up DNA around histones is also called a bobbin. That's more FYI. We already know that the genes that are used to make proteins, which is the blueprint for who we are and what we look like. Okay, we talked about that with our genetics and Punnett squares and all that. Okay, nothing. Okay, so now let's take a look at the DNA replication, the process. Now, definitely look at these videos that are referenced on this slide. So when you go to the actual PowerPoint, pull that up. It will give you an excellent animation of what's involved. They're only like 4 minutes, but it's all that you need to know. It references all of the enzymes like helicase polymerase, the primers, and so on. So you definitely want to pay attention to that, take notes on that, because that's what you'll need to know. Okay. So I'm going to reference it here. I'm going to verbally explain it to you. And then, but definitely watch the videos, and I think you'll do just fine. It's not that bad. Okay, so let's take a look. All right, to begin with, we've got our DNA strand, double helix. Okay, and then we've got helicase. That's this guy right here. All right, so this guy right here, this is an enzyme. These are all proteins: helicase, polymerase, ligase, primase, RNA polymerase. There's a bunch of players. Anything that ends in ASE is an enzyme, aka a protein. Okay. All right, let me just go ahead and take this off. I need to erase this. It's out of my way. All right. Okay, so what's going on here is we got helicase, and it is actually breaking the hydrogen bonds. Okay, it's breaking the hydrogen bonds between each backbone of the nucleotide here, right? Because as I showed you in the previous slides, those dotted lines, those are the hydrogen bonds. So it is breaking. Helicase is unzipping, for lack of a better term. Okay, so what it is creating is a replication fork. It'll say that in the video as well. Okay, it's a replication fork. Now, one thing to note is you've got DNA runs anti-parallel to one another. So you've got like this side strand, and as an example, going this way, and this particular strand is going this way. Okay, so they're anti-parallel. So when you see this, I'm not going to test you on this, but you're going to see it and you're like, what the hell are they talking about? So it says enzyme reads DNA template from a 3' to 5' direction. And then it replicates in a 5' to 3' direction. Okay. So it means that it's reading, this is going through and breaking it in one particular direction, but then it's got to feed off of another when it starts adding nucleotides. But we're not going to worry about that. I'm not as concerned about you understanding that concept because I don't think that's as necessary. I just want you to appreciate and understand the fact of how DNA is replicated. Okay, these are details that I think you'll try to memorize for a half hour, answer 2 questions on an exam, and you'll never remember for the rest of your life because it's not necessary. What does make these unique, though, is at the 3' end you have an OH, and at the 5' end you have a phosphate group. These are molecules. I can show that to you in a second. But anyway, here's what's happening. We've got helicase that's unzipping. So again, we're in the process of replicating. So then what occurs is we've got, as you'll see in the video, we'll have another enzyme that will come in and lock on to this area, because apparently this is the area that's going to be starting the replication process. And then it will bring in a series of these nucleotides. Okay. And basically it's saying, okay, when DNA polymerase is coming in, this is where we want to start. Okay. Then another molecule called DNA polymerase is actually, it's like a vacuum, will suck in these nucleotides that are floating within the nucleus. And these are the resources that you acquired when you had that pork chop last night for protein, because what is protein made of? Protein is made up of amino acids. Okay, you need to get these from this guanine, thymine, cytosine. There's 20 of them, but you definitely need good resources so that your DNA can replicate well. This is part of that good nutrition aspect that we learned in the previous unit. Okay, but anyway, so what DNA polymerase does is it adds nucleotides. So adenine bonds with thymine, guanine bonds with cytosine. So you can predict what nucleotide goes where. Okay, and then there's also then what happens is that there is another DNA polymerase that comes in that goes in the opposite direction. Now this is replicating in one single streamlined event. Okay, it just keeps going and going and going until it's told to stop. On this bottom strand, because it's going in the opposite direction and it's really going in a direction that it can't go, it has to do it in chunks. It's called Okazaki fragments. And again, the video will reference this and illustrate it more clearly than what I'm trying to explain it. Okay, so the same process occurs. You have an enzyme that will go ahead and put some nucleotides here to tell other enzyme, DNA polymerase, hey, this is where you start. And then it will go ahead and lay down the fragment, the nucleotides, into a certain section. And then it releases, and then it will back up and do the same thing all over again, and then fill in the gaps and go back. This is called the lagging strand. This is considered the leading strand because it goes much faster. This is lagging because it goes slower in terms of replication. Now, it's still a very fast process, but relatively it is slower than this. Okay? But definitely watch the video. And again, this is kind of strange because in the video I stop and go, and doing that, which I could do here, but I'm going to have you do it. And then, of course, if you have any questions, you're more than welcome to message me. I'll be more than happy to explain. Super simple stuff. It's not too bad. Go to the next one. Okay, so this is just, again, I don't want to get buried with this 5' to 3' direction. But what designates it 5' to 3', so 5' end, see, this is what's called your phosphate group. The 3 prime, this is what's considered your OH group. And then the DNA, remember, runs anti-parallel. So here on this side, we've got the 3 prime with the OH. And then the 5 prime end is the phosphate group. Okay. So you got one strand of the DNA. This is, of course, your backbone right here. So you got one strand going one way, and the other side of the DNA is going in the opposite direction. And of course, you have your guanine, cytosine pairing up, and you have your adenine and thymine. And of course, the dotted lines illustrate your hydrogen bonds. Okay? All right. Now, that is really it when it comes to DNA replication. And I guess that happens in the nucleus because that's where DNA is. It happens during the synthesis phase of interphase. Okay, so as we try to put the big picture together. Now, what does occur, there are mistakes that do occur. You know, it occurs constantly, but there are enzymes that are read that go back through and read to make sure, it's called ligase, that makes sure that the hydrogen bonds are together and that the right nucleotides are pairing up together. Okay, so that's your check system. Now, remember we talked about that, your checkpoints within interphase. Okay, so here's just an illustration where you got this green chunk thing, looks like Pac-Man, is DNA polymerase. So, of course, you got DNA polymerase that is adding nucleotides. Okay, and then if there is a mismatch, then what happens is that then there's a it's not matching correctly. Then what is going to say, here's the blue and the yellow. Okay, well that's actually supposed to be there. So correct. Let's see if I can see it here. These are all correct. I'm just double checking here. It's been a while since I looked at this, a whopping 4 months. No? So I guess this is illustrating how, I'm not a big fan of this illustration, but it's still worthwhile. This is just illustrating that it is putting the correct nucleotide. This right here is a TED-Ed video. It's about 5 minutes. It's an animation, but it does very good. It's a very nice illustration of showing how mistakes are repaired. So I definitely encourage you to take a look at that. And it points a few of them out. Don't worry about this one because that's confusing to look at, as is that one. Okay. All right, so that is all that there is with DNA replication. Of course, you got some homework that's going to be talking about that in those videos. Yeah, it's really not that hard. So now let's talk about transcription and translation. So that is talking about replicating DNA. So we're talking mitosis, we're talking replicating DNA because of growth or healing your body. That's why DNA replicates. Okay. So now what we're going to be doing is we're going to be talking about how do we transcribe or how do we read the DNA so that we can get that gene that our cell needs for whatever it is that needs to occur with our body. Okay, whether it be growth and development of the child to see what color eyes or what have you. Okay, and so we're looking at or what enzymes need to be created for body function as we're living and breathing every day. Okay, it occurs. So let's take a look at this. So transcription, we're going from DNA to M, which is messenger RNA. Now, if you're reading DNA, where do you think that is occurring? It is occurring within the nucleus. Okay? It has to, because that's where DNA lives. DNA doesn't live anywhere else outside of the cell or within the cell except within the nucleus. Okay, so make sure you remember that. Transcription is occurring within the nucleus where DNA is found. Transcription is transcribing the code that is found on DNA. It is transcribing the gene so that it can make the protein that is necessary for whatever the cell needs that protein for. So here's our key players. Okay, these are the key players. We got DNA, that provides the blueprint. Then you got M. M is messenger RNA. We also have ribosomes. Okay, those are the contractors. That's what translates that messenger RNA when it floats out of the nucleus. It goes through those nuclear pores that you may have seen in past illustrations. And then you've got another molecule called transfer RNA or tRNA. These are the builders. So this is what actually is going to be bringing in the amino acids to build that protein, because proteins are made of nothing more than amino acids, and there's 20 of them. So let's see if we can take a look. So here's what I was talking about, the central dogma of gene expression. You've got your DNA. DNA is transcribed by a messenger RNA molecule, okay? Then it is translated by the ribosome and the tRNA to produce a protein, which is a series of amino acids called created by bonds, polypeptide bonds, okay? They can be very short or very long, and then that protein is off to do something, gets packaged by the Golgi body, and then gets sent out into the body to do whatever it needs to do. Okay? This is about as simple as it gets. Now, obviously, there's many, many details with it, more than we need to know, but this basically is about what we need to know. You wonder if this makes sense to you, you're almost there. There's just a few little details we need to get. Okay. Now, to understand the development of a protein, you need to watch this video. Again, it's a 4-minute video, but it's very good. Okay? Nothing I can say or show you will do as good of a job as this video does. So make sure you watch it. So here we go. We've got DNA replication occurring because we're always replicating DNA. Okay? Depending on what part of the body, you know, say you ate some hot chicken wings and you burn some cells within your lining of your stomach or esophagus or something, so you got to have some replication, you need some new cells. So now you have your DNA. All right, so now we need some proteins, we need some enzymatic action, we need some enzymes created for whatever. So now what's happening is part of your DNA is separating. Again, you've got the helicase, which is opening up a portion of wherever that gene is. That gene is through communication, through molecular reactions, saying, hey, here I am the gene that you were looking for. And then what happens is that there's a molecule, an enzyme that's bringing in, which is RNA polymerase, is now reading this section of DNA. This is one-sided. And reading the code of this gene. It's transcribing, or RNA synthesis, creating this molecule. And then what's spitting out is messenger RNA. Okay, so a molecule just created. This is the code for the gene to make whatever that came from the DNA, a section of the DNA, which is the gene. Okay? So now it floats out of the nucleus, through the nuclear pores, and now we're in the cytoplasm. So here we have this string of RNA, and then we have this big thing called a ribosome. It attaches to the RNA strand. And then atop of it, or somewhere, is another molecule called tRNA. And then what's happening is that this complex of ribosome and tRNA are reading this like a ticker tape. And then when it reads the code, then it spits out an amino acid. And then this ticker tape, then the protein grows longer and longer and longer. And then depending on what its need is and what it is, it goes through the Golgi body, gets packaged, goes form, dictates function, and off it goes. That's probably very confusing, but it's really not that bad. Replication of DNA. Then you have your DNA. Okay, that's why it has it back and forth. Now we want to read DNA, we need to read a gene. That's called transcribing. So we're actually synthesizing an RNA molecule via RNA polymerase. What ends up being created is a molecule called mRNA, messenger RNA. This is the code of the gene. If this is all happening within, all this is happening within the nucleus. Okay? Then it floats out of the nucleus into the cytoplasm. We now are going to be reading this strand of messenger RNA with a ribosome and tRNA, and then it reads the code to create the protein. That's it. That video will show you some more. All right, now a couple of terms. Now, how is it reading and what is it reading to create this protein? Okay, super simple. So it's the genetic code can be broken down into what's called a codon. Okay? What is a codon? Every 3 base pairs, or every 3 nucleotides is being read, so guanine, cytosine, and adenine, that is a code for 1 amino acid. So when this ticker tape, this is the backbone of the RNA, you've got the complex of ribosome and the tRNA on top, and it's reading this. And then what's being spit out is whatever this code is, then what's being brought to the table is the amino acid that this code represents. It's like, oh, there's the amino acid. Then it goes to the next 3, and now it's another amino acid, and there's a bond that occurs between those 2 amino acids, and it keeps going and going and going until it's told to stop. Okay? So it's a codon is a sequence of 3 DNA or RNA nucleotides. Same thing when it comes to with DNA as well. But we're just interested in RNA because you want to know, well, what's being read. So the codon is the genetic code. Now, here's where this mismatching can come into play. Now imagine that there was a mismatched pair, and it did not get caught in the DNA replication, okay? And then the DNA replication happened to be the proto-oncogene that deals with the cell cycle, okay? And say it needed to be replicated. That gene did. We need some more of it for, But say that there was a mutation and it didn't get caught. So instead of this being a G, this would be, say, a T. Well, now it's TCA, not GCA. So this could read as a completely different code for a completely different amino acid. So now the recipe could be faulty, right? So it may not be able to make the protein that is for a proto-oncogene for managing the cell cycle. Now all of a sudden you've got a potential cancer problem, right? Like we talked about with cancer. So I hope that makes sense. That's how mismatching can cause more mutations. Now, it may not even affect it. That protein must still function and work correctly. It may be a benefit in some form or fashion. It may, the mutation could change the color of wings of a butterfly to make it, all of a sudden, it blends in as camouflage with the background of the leaves or something. I don't know. But that's what occurs. It could be a benefit, or it could be a negative. So here's what I mean by these are your codes. So These are your amino acids right here. These are just abbreviations. You don't need to memorize them. You get into biochemistry, they make you memorize them. Okay. These are the 3 letters that make up the amino acids, that codon, if you will. Cytosine, cytosine, uracil. Uracil, you notice that it's reading the RNA. Remember, RNA only does not have thymine. It has the U's. Now, I misspoke when I said, what if it was a T? There is no T in the RNA. But you can see, like, instead of it being AGG, what if it was ACG? So instead of arginine, it would actually make, what's THR? Thramine, maybe? I'm not exactly sure. Okay? So this is what it means. So you've got AAG. There's an amino acid. Then in that complex of a ribosome and tRNA is reading the next 3. It's CAA. So that'd be whatever GIN is. Or GLN. Glycine, maybe. All right. And then leucine, UUA. But you notice that there's a variety of these. You got UUG and UUA both make leucine. And UUC and UUU make pheline or phaline. I forget what it is. Anyway, so that hopefully that makes sense. I wanted to throw this chart in there. You don't have to memorize it. It's not big picture. I mean, it's not details you need to know. Now, some of you may have questioned, well, how does it know where to begin or where to stop? Because within the code, there is a sequence called AUG. It's adenine, uracil, and guanine. And it makes it methionine. So once it sees the code AUG, that means it starts. This is where you start the replication process of creating this gene. Same thing with DNA replications, looking for this right here, AUG. It is the only one. Now, when it comes to stopping, there's 3 possibilities: UAA, UAG, or UGA. It encounters any of those 3 combinations, then it will stop the replication process. That's how it knows. Pretty cool, huh? All right. So but you need to know that? No, but I want you to appreciate it, so at least you understand that just doesn't, to me it seems like it's magical, but there is chemistry to it, and then there's a rhyme or reason, at least what researchers understand. Here's just another, again, I'm being redundant. It's near the last slides. Transcription, translation, what they are. And this is one of those Venn diagrams that just shows you that both have cytosine. These are the amino acids that form a template, both transcription and translation, because this is your RNA. So when we're transcribing, your product of transcription is messenger RNA because you're making that molecule. It occurs in the nucleus. And then, of course, you need that enzyme RNA polymerase, right? And then translation is you're translating messenger RNA. So the product of translation is a polypeptide chain that is a protein. Translation occurs in the ribosomes, so it's in the cytoplasm with the ribosome and the tRNA complex. And then translation uses various reagents to create a polypeptide chain, which I don't know what those reagents are. I did it one time, don't remember. You don't need to know it as long as you know that a polypeptide chain is a protein, which is amino acids bonding together. Okay? That's all you need to know. And then ultimately, again, this is just another illustration showing the exact same concept. You've got DNA. You've got RNA that's reading one strand. Okay, so this is one side, one backbone. Of course, it's reading the one side of DNA. If you know one side, then obviously you know the other. Okay, and then so this is transcription. Then we go into translation. So you've got the ribosome and the tRNA which is then bringing in amino acids. It's reading, see, 1, 2, 3, bringing in an amino acid, which then attaches to a growing chain, and you get the protein. Okay? All right. This is it can get confusing, but this is online, so you don't get all the hand gestures and all that I do and doodling on the board, so I'm hoping this helps. Make sure, well, everybody's been very good about doing their work. Definitely watch the videos. Don't shortcut it. If you don't understand, there's other good videos. There's one video by Drew Barry, I believe, that actually shows real time what DNA synthesis looks like. It's pretty cool, or replication as well as protein synthesis. So, that's something you can check out. Just Google it. But there's plenty of information out there. If you don't understand something or something just isn't flowing, by all means message me, and maybe I can do a video and then I will stop and go it or whatever. I can record it at the same time. I just haven't done it. I haven't needed to. So, okay, guys. Well, I'm hoping you're breaking this in chunks. This is long enough. It's 46 minutes. Good luck.